When you're studying sensory physiology, you may oftentimes hear the term receptive field. So I thought I would take a moment to define what is meant by a receptive field and give a couple of examples. Receptive field is always in reference to a particular neuron that you're studying. So when we think about receptive field, we think about it with respect to the neuron. You could think about the perspective of that neuron. So if I were a neuron, what is my perspective? What can I see out in the world, or what can I sense out in the world? And so you can think then about the receptive field as any part of the environment, typically the space in the environment that can be sensed by that particular neuron. I'll give an example that's a two-dimensional example having to do with our somatosensory system, so our sense of touch. We have sensory neurons in our skin that can detect all sorts of different types of touch. So let's just think about, in general, sense of touch. If you were to touch, let's say, one part of your hand, there would be sensory neurons in your hand that are picking up that touch. It would not be the same sensory neuron that is picking up your sense of touch somewhere else on your hand, okay, or maybe on your arm. That patch of skin, that real estate on your skin, on this part of your hand or your arm, is that neuron's receptive field. And so different neurons may have different amounts of real estate that comprise the receptive field. So for example, I've drawn a receptive field on my arm, and you can see that actually it's common for receptive fields to have two parts, a center part in the middle and a surround, so kind of this donut ring. Okay. I should mention that I've just approximated this. This is not actually a receptive field, but it's a guess based on what I know about our sense of touch. Okay. So imagine a neuron that's sitting right in the middle of this donut ring. It's got, again, the center part of its field and the surround. And a cool thing about sensory physiology is that usually when you touch in the center of that receptive field, that will typically increase, or that could increase or decrease a neuron's activity. But if you touch in the surround, you get the opposite effect. So let's say, for example, just for sake of argument, all of your sensory neurons in your skin will increase their activity if they're touched. So if I touch in the center of my receptive field right here with a pencil, let's say, that neuron is going to be increasing its firing rate. It's going to be firing lots of action potentials. But if I touch in the surround, it will actually decrease its firing rate, fewer action potentials. We call this antagonism or opponency between the center and the surround. Okay. The situation's a little bit more complicated in the visual system. There are actually two types of center or surround receptive fields, but just for sake of argument here, touching in the center, let's say, increases the firing rate. Touching in the surround decreases the firing rate. As I alluded to before, the real estate might be different. So you might already sort of intuitively know that the tips of your fingers are much more sensitive than, let's say, your forearm. And so I've drawn another receptive field on my finger, and you can see that the size of that receptive field is much smaller than this receptive field. So when we study sensory physiology, we need to think about not only the response when you touch different parts of the receptive field, but also maybe how large that area is. In the visual system, it's a little bit different because instead of a patch of skin, we're thinking about a patch of retina in the back of our eye. So like a little, you could think about if you were to spread the retina out like a napkin or this mask here. Let's pretend that this is the back of our retina. You might imagine one photoreceptor having a little patch of retina like so, which is its receptive field. And that little patch of retina corresponds to an area of three-dimensional space. So out in the world, you can map sort of the whole visual space to one particular patch on the retina. Now, that's always changing because we're moving our eyes. So we're able to focus our eyes on different parts of the visual world, but nonetheless, wherever we're focused, let's say in the fovea where you've got lots of photoreceptors, that receptive field is always going to be the same, lying on the same part of the retina. Okay. So I'm hoping that these two examples, the somatosensory example and also the visual example, allow you to understand that the receptive field is an area of environment that, or stimulus in the environment that causes a change in response in the neuron. As you move through sensory systems, that the receptive field, what is required to change the response of the neuron, can get more and more complex. And so that takes us from your photoreceptors having a receptive field of just presence or absence of light, all the way up in your higher visual areas of your brain, neurons that respond specifically to faces of specific people, not just even faces in general, specific faces. And so each way along the way in the visual system, for example, the information that your neurons are paying attention to, their receptive fields are becoming more and more abstracted.